Hi guys, I'm going to give you a quick lecture on proteins today, so follow along with your 2.4 protein notes packet. And I'm going to go faster than normal just because I'm recording this. But this gives you just a whole bunch of different functions of proteins. So unlike carbohydrates where we could kind of say, most of them function in energy, I can't say that about proteins. They have a really, really diverse bunch of functions. We're going to talk in particular about proteins acting as enzymes that run all the reactions in your body, but they're also part of your immune system as antibodies. It's how you recognize foreign things. They carry oxygen in your blood. Structural parts like muscle, hair, and nails are all made of proteins. So their job is just to be sort of structural, to build your body. There's tons that we'll eventually talk through. Today we're going to mainly talk about the structure of a protein. Okay, and so proteins, they're an organic molecule, again, because they have two or more carbons. But they're made of monomers called amino acids. Okay, so the building blocks are called amino acids. Here's a structure of an amino acid. And so it tells you to draw one and then label or highlight in different colors. And so here's the central, you can pause this if you need to, but here's the central carbon. Okay, so all amino acids have a carbon kind of in the middle. And notice there's kind of like then four sides around it. And then you have an amino group, which is NH2, or sometimes we'll see it as NH3 on one side. A carboxyl group, which we should get used to by now, C double bond O, OH, on the other side, okay? Then you have an H, and then on the opposite side of the H, you have what we call an R group, which is like the variable group, which this is actually going to be different in each amino acid. And I always abbreviate amino acid sort of as I'll write AA, that means amino acid. So R doesn't stand for an element. It is like a variable, okay? So you'll see in the picture coming up, there's 20 different amino acids, and the part that you're going to see at where the R is are just different molecular structures, okay? So if you need to pause to draw that and label that, pause, but I'm going to go on. Those are the amino acids, okay? So below in the chart, you can see all the amino acids there are. So there are 20 of them. If you were to count them, there's 20 down here. And when you look at them, what components do they all have in common? So what we were just talking about, they all have a central carbon. They have an amino group. Oh, it has group. They have an amino group and a carboxyl group. So if you look down here, they all have that in common. And the one part that's different, which is the part that's highlighted in different colors, is the R group. Okay, so notice here, they're kind of in three categories. But notice here, you see all of these amino acids. Again, they have a central carbon. They have an amino group, which I said this is the ionized version. It's NH3 plus instead of NH2. And this is C double bond O, O minus instead of OH, but we won't get into the chemistry of that. But you have a central carbon, amino group, carboxyl group, the hydrogen, and then this is the R group, okay? So you can see the R group, is blue in that row, tan in that row, green and purple in that row. And so they're in sort of categories. Remember, nonpolar means no charge or hydrophobic, and so it means it's not soluble. So you can't forget the stuff we learned before. Polar and electrically charged, these things are going to be soluble. And remember, polar has, and all of these things are going to have charges. And so that's what's going to make them soluble. And so we're going to see we're going to just put these amino acids in a big long chain, again, like an amino acid necklace or polymer. And then that chain, so if we put amino acids, if these circles are amino acids, okay, and we link them all in a dot, dot, dot in a big long chain, And let's say this amino acid has a negatively charged R group and this one has a positively charged R group, they're going to be attracted. And if you have a whole bunch of those attractions, this chain will fold up. I'm not going to draw the circles, but the chain will fold up into a globular shape with these attractions between positive and negatives. Okay, so we're going to see when we build proteins, it's a chain of amino acids, but then that's going to have further folding, and that's going to depend on which amino acids are in this chain and if the R groups have charges or don't. The ones that are nonpolar, there's no basis for them to be attracted to each other, and they're going to usually end up in the middle of the protein because they want to be away from water. Okay, so we kind of remember everything in life is in water, so we have water kind of around it. And so the nonpolar amino acids end up kind of in the middle because they want to be away from water. Okay, so for now we're just still kind of looking at the amino acids. There's 20 of them and they all have different names, and you don't need to memorize their names. Just know that some have nonpolar R groups, some have slightly charged R groups, which we call polar, and then these are electrically charged, meaning they're like strong charges. You can see that there's an actual negative charge in this R group and a positive charge in this R group. So here's an example where like those two would be attracted to each other, okay, in a chain, and make the chain fold up. Okay. All right, so we did that. So note then, so I mentioned this in the thing above, carboxyls can be shown in two ways. So I'm just going to let me circle the carboxyl groups in one color and kind of do that on yours too. Pick a color. And notice the two versions you see, there's the C double bond O, OH. It can also ionize and lose this H, C double bond O, O minus. Don't worry about what that means or why. Just be able on a picture, if you see this recognize that as an amino acid and recognize that sometimes they show the carboxyl group as C double bond O, O minus, and then pick another color and you can see the amino group. Sometimes it's, it's NH2. Again, they're kind of drawing it, H2N here, they're kind of drawing it backwards. But that's NH2. But also, you sometimes see it as NH3 plus. It can pick up a hydrogen and a positive charge. Don't worry about why, but just be able to recognize either, when you see a molecule, if you see that, NH anything, it's an amino group. NH3 plus, and again, they just kind of drew it from the other side of the molecule, so it's backwards. But it still has your central carbon, amino group, carboxyl group. It's just that the amino group and the carboxyl group can be drawn different ways. So amino acid chains, I just drew this on the other page, but amino acid chains are linked together. But this is old. We know this by now, I hope, by condensation. Whenever we're combining small molecules, it's a condensation reaction. But when we do that with amino acids, we build long chains called polypeptide chains. Okay, so a polypeptide is just another word for a protein, really. So down here, just write polypeptide protein, and that's what this whole molecule is showing you. It's a protein or a poly. Again, poly means many, and peptide refers to the bonds in between. So these are the peptide bonds. And those are the name, when you do a condensation reaction, you can see this is a polypeptide that has 1, 2, 3, 4, 5 amino acids in that polypeptide chain. And the covalent bond, this is a covalent bond that's strong. Just put it's a strong covalent bond, which means it won't break, under normal situations, okay? And so notice that every polypeptide chain has an amino end, that ends in the amino group, and then if you link them together, the other end ends in the carboxyl group. So we're going to see the carboxyl and the amino group, a water comes out, and that's what bonds them together. Okay. So here's a picture of it. So when two amino acids are joined together, the carboxyl group of the one amino acid is joined to the amino group of the other, and a water molecule is released in the process, okay? And so you can see that here. Here's my carboxyl group, okay? Here's my amino group of the amino acid next to it, and then you can see we took out, it's showing you here, we take two H's and an O, and that comes out as your water. And then that's what causes this carbon to then bond to that nitrogen, okay? So then you can see that down here. Once the water's gone, that forms the peptide bond. So the bond that is formed is a covalent bond. It's a strong bond, and it's called specifically a peptide bond. Okay. When there's only two amino acids that join, it is called a dipeptide instead of a polypeptide. Remember, di means two. So that would technically, this picture here is a dipeptide because there's two amino acids. It's kind of confusing because it makes you think there's two peptide bonds, but there's not two peptide. There's only one peptide bond, but it's joining two amino acids. So they call that a small protein like that. It's called a dipeptide. Okay. So remember, this is all kind of in review. In living cells, proteins are made by, remember, the ribosomes. The little dots in the cell. We learned organelles. And we said that ribosomes do protein synthesis. Okay. And so the sequence of amino acids in a protein, oh, here it's filled in, is determined by the genome or your DNA. So the DNA, what your DNA really codes for, DNA, we all know DNA codes for our traits and our genes are in the DNA, but what DNA really codes for is proteins. It codes for the order of amino acids in all the proteins in your body. And we'll get into that later, but for example, you have brown eyes because you have a brown protein called melanin in your iris. So there's a gene in the DNA that codes for which amino acids to put together. Remember, there's 20 amino acids, and think of them like letters. We just put them in a different order to make different proteins. It's like spelling words. Okay, so we're going to get into that later when we do genetics. Okay, so then let's talk about the structure of the protein really quick here. So we're going to see, let me just show you this kind of overview. So we're going to see that we can build, there's kind of 4 levels of protein structure. So first of all, notice we have, why doesn't it say, oh, let me highlight it for you. So we have primary level, secondary, and you can see this picture in your thing, tertiary and quaternary. So those are the 4 levels of protein structure, okay? And so you'll notice that the primary structure of a protein is just referring to like the little red and blue beads are amino acids linked in a chain, okay? And red and blue is just kind of showing you'll see that some of those will fold into secondary structure, the blue parts. But so every protein has primary structure because it's just the order of amino acids, okay? And then after we kind of get them in a chain, I told you like some of them are going to be attracted, positive and negatives, and they're going to fold. And then there's 2 levels of secondary structure. That amino acid chain can twist into like what we call a helix. Okay, it's called an alpha helix. It's kind of like the picture of starch we did, or a beta pleated sheet. And this one's hard to kind of a beta pleated sheet. It kind of looks like the amino acid chain kind of folds back and forth like that. If you've ever made a fan with a piece of paper where you kind of fold it up back and forth, back and forth, back and forth, back and forth. And so it's kind of like a flat piece of paper. It's like a sheet. That's why it's called a beta pleated sheet. And I kind of drew over that. But so beta, this stands for, sometimes you'll see it listed as a beta sheet or a pleated sheet. And that's the same thing. Okay, so that's like a shape it can fold up into is a pattern. And there's only two types of secondary structure. It can fold up in a helix or a sheet. And so it's just like it's a shape that's a pattern. All right. But a lot of proteins and some, so not all proteins will do this. Not all proteins have secondary structure or will fold up into a pattern. A lot of proteins will just go right from primary structure. Whoops, I didn't want a line there. We'll go right from primary structure to tertiary and quaternary, which is together kind of we call globular proteins. And if you look at the pictures here for tertiary structure, it's the amino acid chain just folds up into kind of a glob. And that's, again, based on those charges between the R groups. I'll draw as dotted lines, like positive ones are attracted to negative ones, etc. But you can see within this big glob, there's like an area of secondary structure kind of within tertiary structure. So that's an alpha helix, that's a pleated sheet within. So we're kind of building on structure here. So it's, if you're building a protein, you build the primary structure first, you put the right amino acids in the right order. Based on their R groups and which ones are charged, etc., that will determine if it, it does secondary, tertiary, how it folds up from there. So when a protein folds up into a blob like that, we call it tertiary structure, okay? And then quaternary structure is the same as tertiary. It's just that there are two amino acid chains. You can see a red chain and a white chain, indicating I have two polypeptide chains of amino acids, and they folded up into the shape. A tertiary only has one amino acid chain. These are the bonds to fill in if you want to pause and fill in the bonds there. And I'll talk a little more about the bonds later. We already talked about the peptide bonds. Those are the ones between amino acids. There are hydrogen bonds that are weak, kind of holding secondary structure together. And then there's all kinds of bonds in tertiary, quaternary. So just kind of write those in. I'll talk those over when we're together. Okay. So just, if you want, you can pause. I'm going to move through this fast, but just to end this video. But here's what you're going to fill in for primary structure. And I already kind of talked about that. And secondary structure, okay? So it's the order of amino acids. Some amino acids are charged or polar. Remember, polar means slightly charged. But if it has a big charge or a slight charge, they're gonna attract and fold up. So I'm moving, but you can pause the video now if you need to fill that in, okay? And then tertiary and quaternary structure, again, I'm going to move quick. I'm going to end this, but you can pause this to fill in your notes. An example of quaternary structure is hemoglobin, found in RBC, stands for red blood cells. And it carries oxygen in your blood, okay? I'll talk a little bit more about the bonds in this when we are together next class. So if you need to fill this in, you may pause. And then the last thing to fill in, these are just two examples. Keratin is an example of protein found in your hair, and it's an example of, you can see the alpha helical structure, okay, which is a type of secondary structure. And then silk is an example of a protein that has beta pleated sheet. And this is a little better. You can see what a beta pleated sheet kind of is. It's sort of like, it's the chain of amino acids that are just folding back and forth on each other, and they kind of kink like that. That's a beta pleated sheet. So again, pause if you need to, go back to fill those in. I think that, and then here's the last part. It talks a little bit more about hemoglobin. And I believe we are almost at the end. Again, just pause it, fill this in, and we'll do your best on the homework, okay? And then I'll talk about anything and help you with it next class. All right, so denaturation. Denaturation is when a protein loses its shape, which often leads to a loss in function. Okay, so when a protein falls apart, we call it denaturing. And some factors that can cause denaturing are heat, strong acids, or strong bases. Okay, so a change in pH. Acids and bases are measured in pH. These factors cause denaturation because they disrupt the weak bonds, okay? Not the strong covalent bonds of the primary structure. That's what that's supposed to say, but I think you have that. Okay. Are we missing something there? Just fill in what you can, and then again, I'll kind of quickly go over this next class if there's any problems. And I think that's it. I was going to talk a little bit about hair, but I'm not. So I'm going to end the recording there. Go ahead and fill in your notes, do your best on the homework, and you can also refer to 2.4 in your textbook for help. Okay? I'll see you next class.